manuscript to the faculty board on January the 21st. After the closed defense on March the 10th, the examination board have decided that the PhD candidate is admitted to the public defense. So I'm proud that I can present the members of the examination board. So I will start with Dr. Sophie Mantelen of the James Hutton Institute in Scotland. Professor Bartel van Hoom of the Faculty of Science, Department of Biotechnology and Bioinformatics of Kent University. Professor Sophie Hoormachter of the Faculty of Science, also of the Department of Plant Biotechnology and Bioinformatics, also of Kent University, of course. Professor Geert Smant of Wageningen University. He is after me, I think. Yes. And I will end, oh no, I forgot the most important person, the Secretary of the Examination Board, Professor Ismaker of the Department of Crop Protection of our faculty. We have two promoters, Promoter John Jones, also of the James Hutton Institute, and Professor Lieve Hees of the Department of Molecular Biotechnology of our faculty. My name is Geert Gazaert. I'm the chairman of the examination board. I'm very easy task this afternoon and must facilitate this meeting. So the title of the PG manuscript is Functional Analysis of Globodera pallida Sprysac Proteins. So, Mrs. Yuan, we are very curious to hear about the main results and topics of your work. So you know you have 30 minutes, 35 minutes maximum to do it. And I would say the floor is yours, so you can start.
University and the James Hutton Institute in Scotland. And my PhD was to investigate the function of Globodera pallida Sprysac proteins. Before I show you my results, I would like you to join me welcoming the two protagonists of my story. The first one is called the potato plant, with the scientific name Solanum tuberosum. This plant has been grown in more than 100 countries and has been ranked as the number 4 most important food crop in the whole world. The second protagonist is quite tiny, but sometimes you see them in a shape of a thread, and sometimes you can see them in the shape of white balls. They are the same thing. And they are called the white potato cyst nematode, with the scientific name Globodera pallida. This nematode exists in almost all potato growing areas, and it's ranked as the number 2 most influential plant parasitic nematodes in the whole world. This nematode has a very interesting life cycle. It starts its life in the soil, hatching from the egg, looking for a plant to penetrate into the plant. And then the most interesting part of this nematode is that the sex differentiation is heavily controlled by environmental factors such as nutrition supply. If there's plenty of food, the nematode will decide to become a female by forming a very large feeding site. If there's not enough food, it will decide to become a male, like here, and it regains this very slim body shape and exits from the root. The feeding site of this nematode, we call it syncytium. It can consist of like hundreds of plant cells. They look pretty tight, like all very broken. But this nematode can somehow keep these broken cells alive for a long period of up to 8 weeks. So you can imagine that this nematode and this plant are having a very fascinating relationship. But unfortunately, they did not end up living together forever, like happily ever after. What you see most often in the field is like this. The potato plants become so thick that it's like yellowing stumps, and sometimes the plant just dies off entirely because of the nematode. This is so sad, and it's kind of tragedy. Unfortunately, it didn't get the attention of policemen or social workers, as you can imagine, but a group of people, very clever people, called scientists, they looked into this case, and they made pretty fair judgments. What they point out first is that potato plants are not as useless as you might thought. Of course, plants cannot run away at all from pathogen invasion, but they have something called innate immunity. When pathogen or nematodes invade a plant, the plant first recognizes this pathogen as PAM, pathogen-associated molecular peptides. By its surface receptor, we call it PRR. And then a signaling will be provoked, so the plant has some immune response. And this layer of response, we call it PAM-triggered immunity. It's usually sufficient enough to ward off the majority of pathogen attack. Aggressive pathogen can secrete some proteins called these purple stars. They can secrete these kind of effector proteins, we call, and these proteins can interfere with this plant defense. So plant defenses are inactivated. But plant evolves with a second layer of plant defense. Plant can come up with a resistant protein, and this protein can recognize these yellow stars. And then it has an even stronger plant defense, which we call effector-triggered immunity, ETI. In this case, we call these yellow stars AVR proteins. So bear in mind that ETI response always involves a recognition between AVR protein, pathogen AVR protein, and plant-resistant proteins. And the most typical symptom of ETI response from a plant is the cell death. Scientifically, we call it hypersensitive response. This is a very strong plant immunity system, where the plant can sacrifice a small part of its cells to protect the whole body from being destroyed. This is the plant side. Okay, Candida, the nematodes, being so destructive. And why? Because they can also, like other pathogens, they can secrete effector proteins. Effector proteins from nematodes are mainly synthesized from gland cells, and they can secrete it into plant cells by a very special structure. We call it stylet in the mouth region here. when the secretions delivered into plant cells, these proteins can reprogram plant cell development, and then it will have a very fast feeding site to obtain nutrients. And in the meantime, it can also cause disease. Okay, to look for effector proteins from G. pallida, the scientists decoded the genetic information of this worm. Genetic information is basically DNA. DNA lives in the center of the cell. DNA contains genes, and it can be further transcribed into messenger RNA. And in pallida, as in many other organisms, messenger RNA can be further translated into proteins.